This protocol shows two distinct ways of detecting NKG2DLs on the surface of AML cells. The technique provides a fast, user-friendly staining method to detect all known and possibly unknown NKG2DLs. This method allows for separation of leukemic stem cells from bulk AML cells, which makes it possible to further characterize these cells. Begin by thawing the biotin and the NKG2D fusion protein tubes at room temperature. Quickly spin down the NKG2D FC powder, then add 500 microliters of PBS to reconstitute the powder and mix thoroughly using a P1000 micropipette. Add 100 microliters of the NKG2DL fusion protein to a biotin tube to obtain a final concentration of 10 micrograms per milliliter and mix the solution thoroughly with a P100 micropipette. To thaw the AML cells, remove the cryovial containing primary AML from the liquid nitrogen storage and immediately place it in a 37 degrees Celsius water bath. Gently move the tube back and forth in the water, allowing the contents of the vial to thaw until there is only a small ice crystal left. Immediately transfer the thawed cells into the medium-containing tube and rinse the vial using 1 milliliter of medium. Centrifuge the cells at 300 times G for 10 minutes and discard the supernatant without disturbing the pellet. Wash the cells with 5 milliliters of RPMI medium containing 10% FCS and repeat the centrifugation.